Hello, I'm Eleni Birli. I'm a Bioassay scientist 3 at Cellomatics Biosciences. I joined the team of Cellomatics 3 years ago. Asthma is a respiratory disease marked by chronic airway inflammation and airway hyperresponsiveness. Despite ongoing research, there is still no effective cure, and this is partly because this disease is characterized by deregulated communication between the airway epithelial and the underlying mesenchymal compartment. Understanding and targeting these kind of interactions would allow us find better treatments, and this is where the 3D in vitro co-culture models play a significant role. Modeling asthma in the lab can allow us dissect the different cellular mechanisms driving inflammation and remodeling. Traditional models used so far, like the 2D monolayer cultures, can give us some information about cell behaviors, but do not allow cell-cell and cell matrix interactions to occur naturally, which is something very common in diseases like asthma, where different cell types can influence each other. At Cellomatics, our basic model uses the airway epithelial cells, which are called Bs2b, on a semi-permeable membrane to represent the airway lining. Lying beneath are the smooth muscle cells to represent the muscle layer. This setup can allow the direct and indirect interaction of these two layers. In our tree culture model, we further add freshly isolated PBMCs to represent the immune system. PBMCs bring an immune component to the system, which help mimics the inflammatory environment seen in asthma. This allows us to study the inflammation and cytokines production upon stimulation with viral mimics, which are key triggers in asthma. To induce inflammation at Cellomatics, we use poly-IC, which is a viral mimic, and LPS, which is a bacterial endotoxin. They both act by activating toll-like receptors, enabling us to study the inflammatory cascade in detail. To ensure our model reflects the physiological conditions, we induced a contractile phenotype in the smooth muscle cells by culturing them in low serum medium, supplemented with growth factors. This was very important because we wanted to mimic the contractile phenotype of the cells seen in asthma. In order to verify this phenotypic shift of the cells, we checked the expression of some well-known contractile related genes, like the myosin light chain kinase, the alpha smooth muscle actin, and the calponin-1. Interestingly, the results showed the increase of the expression of all these three contractile related genes verified the contractile phenotype of our cells. This confirmation was essential to ensure that our experimental model accurately reflects the cellular behavior which is associated with asthma remodeling, thereby increasing the biological relevance of our results. Then we went to assess the inflammatory signaling using a Luminex multiplex assay to quantify different cytokines and chemokines which play a key role in inflammation. More specifically, we studied the production of IL-6, IL-8 and MCP-1, which have a significant role in inflammation and active cell recruitment. The Luminex results revealed that there was an inflammatory response after the LPS stimulation, especially in our tree culture model. This response was even stronger than the monocultures and co-cultures, highlighting the importance of the multicellular interactions and suggesting active signaling between the different cell types. To investigate the mechanism underlying the cytokines production, we pretreated the cells with the IL-1 receptor antagonist. Interestingly, IL-1 array effectively blocked the LPS-mediated cytokines production. This result showed two things. First, that the IL-1 signaling plays a significant role in the inflammatory cascade, and second, that the cytokines production was not only affected by the LPS stimulation, but also was dependent on the downstream IL-1 signaling. Our system offers scalable and human physiologically relevant platform for screening anti-inflammatory and bronchodilator compounds. We can assess both the efficacy and the mechanism of action in a controlled and validated setup. Compared to animal models, our tree culture model is cost-effective, obviously more ethically sound, and it can be easily scaled up from medium to high throughput screening. This accelerates the screening to find more effective treatments and even helps to identify new targets. By using patient-derived cells, our model could be tailored to reflect individuals' inflammatory profiles, enabling personalized treatment. This approach could help identify more effective treatments for different asthma phenotypes. If asthma is your area of interest, we would love to hear from you.